Okay. Hi, everyone. Thanks for coming. Watch our talks. I'm Bruno Perez. I'm a geneticist for Hendrix genetics, and I'm here to present a small part of the work that we've been doing within the GENE-SWitCH project. And I'm going to talk about including function annotation into genomic prediction models for the prediction of gene expression in pigs. Okay. So introduction, pretty straightforward. Methylation has known effects for regulating gene expression patterns, although the full extent of these effects is not completely clear. But yeah, this is known. Methylation affects gene expression. And so differentiated cells may inherit unique DNA methylation patterns that can be linked to tissue-specific gene expression. So this, in practice, would mean that functional annotation methylation from different time stages or tissues could have different benefits when for performance of prediction in either gene expression or phenotypes ultimately. So our hypothesis there was, okay, so probably using methylation information as prior knowledge in a genomic prediction model could help to improve the prediction accuracy if the intent is to predict gene expressions. So basically our objectives was to include the methylation information as weights in genomic prediction models for gene expressions in pigs. So how did we achieve that? So the data we worked on, well, we used whole genome sequence, so we had 300 pigs sequenced. It's not imputed, it's a real sequence. So 100 landrace, 100 duroc, and 100 large white, different populations. The size of the logos for the other companies, they're small, it's not intended. It's just, I receive it like this. So 3 different populations, 3 different breeds. For phenotypes, we used gene expression from 10 different genes. And we could have also ran it for all the annotated genes. But then what we actually did, we used these 10 genes that were selected as candidates by the group in Spain, also from GENE-SWitCH. And they had previous results that indicate that they are, well, markedly controlled by methylation. So we had gene expression on these animals for liver tissue, duodenum, and muscle tissue. And this was measured around 6 months of age. For the function annotation, so the methylation information that we had methylation states. I'm going to explain a little bit more on that. But this was the relevant part to there is that it comes from a different large white population. It's not the same large white population that we had gene expression and whole genome sequence for. Well, also important to say that we had methylation states for the same tissue, so liver, duodenum, and muscle, and actually 3 time stages. I think Elisabeth just presented. Yeah, we had embryo stages and newborn stages. Okay, so the thought behind selecting the regulatory region. So what we had whole genome sequence, and when we started looking at this, we actually used whole genome sequence as it is. But then results were not really conclusive. So what, as a next step, we decided, okay, so what if we narrow down our map, reduce dimensionality, and look at the parts of the genome that should contain promoter functional elements, and then we should be able to see something, if there is an effect, if we can do it. So then we subset from the whole genome sequence only SNPs 2K upwards the coding strand, the TSS, and 0.2 KB within the gene region. In total, we had around 60K SNP variants remaining, and on average 3 SNP per regulatory region, but it ranged between 1 and 12. On the methylation, so the methylation variable that we were working with, it ranged from 0 to 1. 0 being not methylated, 1 being fully methylated. We actually had more tissues than what I'm presenting now, and 3 time points. So 30 days post-fertilization, 70 days post-fertilization in newborn stage, So these are 21 combinations of tissue time points. So what we did, for the weights, to derive from the methylation variable to a weight that we could use and for prediction, for weighting the relationships in the GRM, we just inverted this relationship and assumed a linear relationship between methylation and gene expression. So basically, now on the weight, 1 means full effect, 0 means no effect. Oh, yeah. And then in this formula, meth is the methylation states per locus on a given developmental stage and tissue. We also, we didn't use exactly the methylation at the locus itself. We actually used methylation, the average methylation around the locus, mainly because we wanted to avoid the non-segregating variants in a way that the results for the methylation, if you only observed one allele, could be biased. Just for illustration, this is how the methylation patterns looked for each of the tissues. Quite similar. If you take liver, it's a little bit different, but they differ really on the right tail, the extreme values. And indeed, they look similar, and I think the main differences across them, if you calculate correlations, they're quite high, but the main differences are in specific regions on the genome. So predictive scenarios that we tested. So first, of course, we used the GBLUP unweighted GRM as a baseline. And we also used weighted GBLUP, including methylation information, so weights on the GRM coming from the liver, duodenum, and muscle in the, 3 time stages. And then, the GRMs that we built, we assumed that we had 3 different breeds. So we accounted for the breed-specific allele frequencies according to Weentzsch. We had 2 validation schemes. So first we thought of a cross validation. So we designed a 5-fold cross validation. Basically, we used a k-means cluster within each breed just to separate any family ties that we might have. Then we reallocated them, combining with all other breeds in a way that we would have the same number of animals for every breed, either in the validation and in the reference. And we also designed in a crossbreed prediction, in which we always have on a reference 2 breeds and the remaining breed in the validation. Okay, so I'll guide you through the results. Start with the 5-fold cross-validation. So in this scenario, I'm presenting predictive accuracies for the gene expressions on liver, right? And just to help you through it, so if we assume that the methylation might help explaining gene expressions, so then we can also assume that if we have methylation in liver, this should have better performance than either using, at least than the unweighted, but it could also outperform the methylation from using methylation from other tissues. So the results, what you see here is the percentage relative difference between this model and whatever model it is, and the unweighted. And one thing I wanted to stress, maybe, it's pay attention to the first gene and the last gene, so the ones that are negative, and IGF2 and NUDT2 in the middle, because they have similar patterns between them. So what we can see is, for 7 out of 10 genes, using liver methylation to explain gene expressions on liver using SNPs yields a better performance than the unweighted model. Although it's a small difference, but it's there. Now doing the same thought with duodenum. So now we would expect the green bars to be up, and now we see a more marked difference. So again, if you look at the first and the last, they are still negative. It's a pattern that we observe across the analysis. And IGF2 and UDT22 also positive with really marked increase in performance when you use methylation from duodenum, either developmental stage. Then for duodenum we observe 6 genes out of 10. The weighted model was better than unweighted. Same thing for muscle. Well, you already understand the thought. So now for muscle, the results are not as marked, let's say. There's more variation, but still, first and last genes still negative. IGF2 and UDT22 are still positive, and when we use methylation. And then we have 5 genes out of 10 in which using methylation in your model outperforms the unweighted, the traditional GBLUP. Okay, now switching to a crossbreed validation. I'm going to show just one of these scenarios using landrace and Duroc as a reference and predicting on large white, but here the picture is slightly different. So basically, what you're seeing here is not percentage of difference, it's the predictive accuracy absolute value. And you see duodenum liver muscle on top, so these are the gene expressions that you're trying to explain. But you see that the pattern completely deteriorates, so there's pretty much no pattern observed, so it's all over the place. So as take-home messages, so using methylation as functional annotation yielded strongly trait, in our case, gene-specific results. For the cross-validation using 3 breeds, we observe a slight positive trend, and I'm going to explain a little bit of the reasoning, why we think this happens. But then for a crossbreed prediction, predictive accuracy gets really deteriorated. And if you go to some human studies that did a similar task, they have the same conclusion, trying to predict across populations, so African, European, or American European. It really deteriorates the power of predicting gene expressions and using methylation. So it looks, well, performance seems dependent on the effect of the methylation, and it makes sense. The contribution that methylation might have to predicting with SNP the gene expression is probably dependent on how methylation affects that gene. But not only that, so if you think of genomics, it's quite static for the whole life of the animal, of course. But if you go to methylation and gene expression, then you have a more dynamic relationship. So the time of measurement for the methylation and for the gene expression will probably have a big effect on that, on the predictive accuracy. So what we can say from that is that some of the genes in which we tested, and probably throughout the whole genome, for these genes, methylation, either on embryo or newborn stages, might have limited contribution to the gene expression, either because they don't play a part shutting down or activating that gene expression there, or just because the time points that you're analyzing, you lose that trigger, that switch, right? So for the future, which at the time was the future, now that's what we're doing now. So in practice, we have multiple layers of functional annotation, not only methylation. And it looks like from other studies that we've seen, when we use one layer of functional annotation as prior knowledge, results are often really trait specific. In our case, gene specific, but trait specific. So what it looks like, blending all these layers using any method, it would be detrimental to really capture the benefits that functional annotation can bring to genomic prediction, either for gene expressions or phenotypes. And thinking on the ultimate goal, which is apply this method, include functional annotation into genomic prediction models for breeding purposes. So, in the scope of a breeding program, then there we have a multi-trait approach. So if you have a really strongly trait-dependent performance for some model, then you have a problem, right? Because I think no geneticists want to lose accuracy in detriment of gaining accuracy on other traits. Well, maybe. And finally, just what we're working on now. I think there were already the Fed scores were already mentioned during the conference. So this was published by Xian 2019. It's basically a framework for scoring different SNPs and according to their functional relevance, so according to how much percentage of variance they can capture across your functional maps and phenotypes. And they can be easily further used as prior knowledge in genomic prediction models, either in Bayes-R, Bayesian models, or GBLUP. Yep. With that, I'd like to thank other partners and open for questions.